Children, this is Aniruddha, Mount Litra Z School Jhansi. In the previous section, Biology 12th class book of NCERT, we were talking about the contraceptive methods and we had talked about the surgical method of contraception, that means vasectomy and tubectomy, and their side effects also. Now today we are going to talk about medical termination of pregnancy, or it is also known as MTP, or it is also known as induced abortion. In India, this medical termination of pregnancy was legalized in 1971, but it has certain restrictions, okay, just to prevent the misuse of this particular practice. In India, there is a concept that the parents need only the male child and they do not need the female child. That's why most of the time the female is illegally killed inside the womb of mother, or medically the female is terminated. Okay, the pregnancy is terminated. Just to stop this illegal malpractice and the discrimination among children, male or female, this practice had certain restrictions even after the legalization. Actually, the major thing why MTP is used, just to get rid of unwanted pregnancy. It may be just because of failure of contraceptive methods which are being used, or it may be just because of unprotected intercourse, or sometimes in the cases of rapes. And the second thing, sometimes the child who is developing inside the womb of mother becomes very dangerous for the life of the mother, okay, just because of that harmful effect of fetus. This MTP, or the medical termination of pregnancy, is done. Or medical termination of pregnancy can only be done within the 12 weeks of pregnancy, that means the first trimester. But in the second trimester also it can be done, but it is very riskier. Okay, it can be riskier for the female, or it can be riskier for the reproductive life of that female. So this is all about the MTP, medical termination of pregnancy, which is only done by some practitioners, medical practitioners or experienced practitioners or nurse. The second thing is sexually transmitted diseases, which we are going to discuss today. Sexually transmitted diseases, that means the diseases which are transferred from sexual intercourse or sexual contact. These are also known as venereal disease, or also known as reproductive tract infections. You must have listened the name of certain STDs such as gonorrhea, syphilis, genital herpes, AIDS, hepatitis B. These are some common diseases which everybody hears. Hepatitis B and HIV can also be transferred from one person to another by the use of certain needles, surgical instruments. Okay, if it is being it has been already used by some infected person, or just because of transfusion of blood also, it can be transmitted. It can be transmitted also from the fetus to infected mother, okay, by the blood fusion or something else. There is one concept that HIV and hepatitis B can never be completely cured, but this is not true, except genital herpes, except genital herpes. All the sexually transmitted diseases such as HIV, hepatitis B, they can completely be cured. They are curable, but if they are detected in the earlier stage and treated properly. If the detection of HIV and hepatitis B is done in the earlier stage and it is properly treated by experienced doctor, it will completely be curable. If the timely detection is done and the treatment is done, there will be no complications in the later part of the life, okay, and there will be no problem in births or the reproductive health of that male or female. Now, how, as you know, that prevention is better than cure. There are so many diseases which are very dangerous, okay, however they can be cured completely, but the damage they cause to the life or the damage they cause to the body can never be repairable, can never be returned. Okay, so it is better that you prevent the disease. That is why it is said prevention is better than cure. If you cure the disease, it will definitely damage some part of your life, some part of your body, some part of your any organ, and you cannot gain again the same power, the same strength in that organ, in that body. So it is better that you prevent the disease. How can you prevent the STDs? Avoid sex with unknown partners or multiple partners. Always use contraceptive methods, which is condoms. Okay, the barrier method. Other methods are not effective in this STDs. Okay, so only the barrier method is the most powerful method, which gives a kind of ability that male and female partner, even after the mating, even after the intercourse, their body fluid does not transfuse. And if you have any problem, any doubt, if you think you may have this disease or you have intercoursed with multiple partners, always go to a qualified doctor and clear your doubt, or take a test. That will be better. This is all about the sexually transmitted diseases. Or if you are found with any sexually transmitted diseases, always, if the diagnosis is done, start the treatment as soon as possible. Now the next thing is infertility. Infertility means unable to produce or unable to reproduce children. In spite of unprotected sexual contacts, or maybe just being sexually healthy, still there may be infertility. It may be the problem of male, it may be the problem of female. It may be just because of physical problems, congenital problems, that means the genetic problems. It may be just because of certain diseases, it may be just because of the side effects of certain drugs, it may be just because of immunological problems or psychological problems. That is why reproductive health, the definition of reproductive health has been given in such a manner that it is the complete well-being of a person, whether it is physical, mental or social. Okay, because they all have effect or impact on infertility, on the reproductive quality of a male or a female. So there are so many techniques which now have been developed for those couples who cannot produce children. They are known as assisted reproductive technologies, or in short they are known as ART. Number one is in vitro fertilization. You must have listened about this. It is IVF, or it is also known as test tube baby program. In this, what happens, from a male sperm is taken, or from a female ova is taken. They are collected, and in laboratory they are fused together to form an embryo. In laboratory, the zygote is developed up to the stage of 8 cells, that means early embryo, and in that stage it is transferred from tube to fallopian tube of a female by any of the methods. It can be ZIFT, that means zygote intrafallopian transfer, that means the zygote doesn't develop into the embryo, but in the zygotic stage that cell is transferred directly into the fallopian tube and the further development takes place inside the female. And it may be inside the tube the zygote is developed till 8 blastomeres or 8 cell stage, that means till early embryo, and that embryo is transferred directly into the uterine cavity of a female, that means inside the uterus. It can be transferred, zygote can be transferred to fallopian tube, from where it will develop into the embryo, and naturally it will be implanted inside the female's uterus. And in second method, in IUT method, that means intrauterine transfer, inside outside the body, in the test tube, zygote is developed till the early embryo, and then embryo is directly transferred into the uterine wall, uterine cavity of a female for the complete further development. There is one more technology known as GIFT, gamete intrafallopian transfer. In this, what happens, from a donor gamete is collected, That means a female gamete or female ova is collected. If the lady is not able to produce ova inside her ovary, then ova is collected from a donor and it is transferred inside the fallopian tube of another female. And intracytoplasmic sperm injection, we all know, we have read in chapter number 3 that sperm goes to the ovum after the release in the vagina of a female. It reaches to the ovum and it dissolves the wall of ovum, the outside wall of ovum, and then enters inside the female's ovum and fertilizes the another chromosome. Actually, if there are certain problems with the sperm and it is not able to dissolve the wall of ovum, then some specialized machines are used, procedures are used, such as gene gun or a kind of cytoplasmic gun is used, through which the sperm is directly injected inside the ovum. There is artificial insemination. So many times in the infertility cases, male is unable to produce healthy sperms. Okay. By this, actually what is done, semen is collected from the donor and it is artificially introduced to the vagina or into the uterus of a female. Okay, here this chapter finishes, and in next chapter, the next chapter is the chapter number 5th, that means the chapter of genetics, which we will start next time. Thank you very much. Have a nice day.